Hello, everyone, and welcome to today's webinar, Minicircle DNA, a new material for CAR-T and AAV vector particles. Its development and analysis presented by Dr. Martin Schleef, founder and CEO of PlasmidFactory. I am Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by SciEx. To learn more about our sponsor, please visit them at sciex.com. Now let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing that presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Martin Schleef, founder and CEO of PlasmidFactory. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Schleef, you may begin your presentation.
Thank you, Susie, for your kind introduction. Today I will talk about minicircle DNA. Minicircle DNA is new. People know about plasmid DNA, and minicircle is, let's say, one step further. Minicircles are like plasmids. They are circular, but they contain less than a plasmid. For example, they don't contain the origin of replication and the antibiotic resistance marker. Let me start with explaining more about plasmid DNA. The structure of plasmid DNA in textbooks is like a perfect circle, but in reality they are looking different. They look a little bit like the cable of a telephone. In the panel, the upper left picture, A, this is a super-coiled monomer of a plasmid. This is how it should look. It's a perfect plasmid, it's intact, and it works. If it is cut, for example, by a restriction enzyme, or destroyed by whatever kind of lab accident, it is a linear molecule. It's not condensed anymore, and this means both strands are damaged. If only one is damaged, it's still some kind of circle, but it's not supercoiled anymore. It's a relaxed thing in the upper row on the right side. And if they are dimeric structures, two plasmids in form of one molecule, this is the lower row where you on the left side can see the dimer supercoiled, in the middle the linear damaged dimeric plasmid, and on the right the relaxed dimeric form. To prove this, we made an atomic force microscopy picture, and there all different forms can be seen, and they are really condensed as this is shown in the picture. And since we are manufacturing these plasmids for applications like gene therapy or simple transfections in research, it is very critical to ensure that the plasmid is intact, because this is directly influencing the success of the research or of the intended work. And if the plasmid is damaged, it simply doesn't work anymore. This is why we try to purify only those CCC forms, the condensed coiled forms, upper row on the left side. And I have an example with me on the next slide, which is the type of picture that we all know from the lab work. On the left side, there is a agarose gel electrophoresis picture. There is a DNA marker on both sides, but there are lanes 1, 2, 3, and 4. And it is interesting to have a look to lanes 1 and 2, where you can see a big band. This big band is the supercoiled form, the one that we would like to isolate, but there is something above, and this above is a double band consisting of the dimeric form, so double size, but also of this open circular damaged form. The linear one is not presented here. And lanes 3 and 4 show a plasmid that is perfectly purified and only consists of the condensed plasmid, the CCC form. And if you look to these upper bands, which you can see in 1 and 2, you don't see them in 3 and 4 anymore. You only see a little bit of one of both, which is the supercoiled dimer, but the open circle one, the damaged form of the plasmid, is gone. And this is important because now the quality of 3 and 4 is representing the highest possible quality of a plasmid. And on the right side, you can see the capillary gel electrophoresis of such a DNA, where we have one big peak where you can see the monomer of the supercoiled plasmid, and you see a small peak which is the CCC dimer, so both are intact, simply double size is the small peak, and where the open circle should be, there is no peak, because in this purification technology we simply don't have it. And this is the quality that people like to use for all gene therapy applications, but also for making molecules in the process of producing gene therapy products, because they have high potency, they are effective, and they don't produce defect material for subsequent further work. And one field where we are delivering starting material to is the field of making gene therapy viral vectors. And these gene therapy viral vectors are quite famous in these days. There is so-called lentiviral vectors or AAV viral vectors. And I would like to focus today on AAV, which I will do on the next slide. AAV viruses are made with different types of transfection of producer cells. The producer cells are transfected with different genes, all necessary to make the AAV particle. And these genes are spread on different plasmids. The typical technology of the past was putting these elements on 3 different plasmids for the helper, for the packaging, and for the gene transfer element. And these 3 all together have to be used to co-transfect a manufacturing producer cell, and this producer cell is then making the viral particle. We have something which we worldwide exclusively licensed in from the German Cancer Research Center in Heidelberg, Germany. This is something which is quite helpful for all people doing this triple transfection, because it's a co-transfection simply. This means there is one plasmid which carries the transgene in green in this picture, flanked by ITR sequences, very important for making the AAV, and all other genetic elements that are required to make the AAV are on the other plasmid, the red and blue. There is some other elements, some E genes and so on it, and this other plasmid is quite big. It's approximately 20 kb in size. All elements are there, and only these 2 have to be co-transfected, and together they cause the production of AAV viruses within the cell. They come out of the cell, and they are purified further in the manufacturing technology that is applied to get these AAV particles isolated, and they are the gene therapy vectors that are presently used to cure certain diseases with success. And the plasmids are the starting materials for doing this, and have to be manufactured in a high quality, for example in the CCC form that I have shown you before. There is different types of available plasmids carrying the helper and packaging functions. So just some examples here. There is Cap2 always, with one exception, and different serotypes, Cap1, Cap2, Cap3, and so on. So this is available with or without red fluorescence protein, where you can monitor if the transfection works in the cell. So these different features are there, and since this is something which is frequently used by the industry and academic labs, this is something which we have pre-manufactured in a quality that is more than sufficient for doing this successfully. And this is just an overview. Important is that you can see that these plasmids are really quite big because they have to carry all the genetic elements that are used to make these AAV packaging in the cell. The next slide, I can show you two examples of these two plasmids. On the left side, called PDP2, is this plasmid carrying all the helper and packaging functions. There is a RAP2 and a CAP2 in blue and yellow that you can see. This causes the production of AAV particles with a serotype of CAP2. This means that this has a specific tropism that leads the virus to go to a target tissue. This is why people really love this AAV, because it's going to the target, and you can select another serotype to target other tissues. On the right side, there is a reporter gene, just as an example for you to see that it's possible to combine the PDG on the left side with a transfer plasmid on the right side, and this transfer plasmid contains the gene green part is a GFP, just a GFP as a reporter to be able to see that the transfection works and that we are able to make viral particles of the AAV type that successfully can transfer the GFP into the tissue that should be treated with the AAV. I would like to explain that unfortunately it has shown that the system of making AAV is not perfectly working in all cells. It happens that the part outside the flanking ITR areas of the plasmid, where the antibiotic resistance of the plasmid is located in the origin of replication, only elements that are needed in E. coli in manufacturing plasmids, are packaged instead of the part that should be packaged, which is the GFP that I have shown you on the earlier slide. And what we did in collaboration with colleagues from Cologne and Hanover is that we analyzed these viral particles, and it turned out that, of course, as expected, within the viral particles there is a lot of GFP. So the majority of these viral particles contains the GFP in a quantitative PCR assay. It has been analyzed from AAV preparations that have been purified with all different types of technology. There is 3 sets of columns, vector production 1, 2, and 3. And they have been purified with affinity chromatography, with gel filtration, and so on. And the gray columns that you can see show that the GFP is present all the time. And this is what, of course, we wanted. But we do not want to see the orange columns, because the orange columns show that the ampicillin resistance gene element, which is outside the area that should be packaged by these AAV vectors, is packaged, although it shouldn't be, because it's on the other side of the ITR elements, and this is what, of course, no patient wants to be treated with, and it shouldn't be there. There is orders of magnitude between this, but as you can see, it's there and it shouldn't be there. And the idea is now how to avoid that these wrong sequences that are not treating, they are just a contamination of an AAV preparation, how to avoid them in such a production, especially if it's a pharmaceutical production of AAV particles. You can see in this summary that this backbone sequence does not contain the EGFP, and in case of single-strand AAV vectors, it is only 0.5 to approximately 3%, but in self-complementary ones, it is up to 26%, and this is a quarter of the total production of such a vector. And this, of course, should be avoided, which also can be seen in the guidelines for medicinal gene transfer products that I mentioned in this overhead. So the idea is we wanted to avoid these wrong sequences in the AAV viral particles. And what helped us was that we, since 15 years, developed a system which is the minicircle that I mentioned in the beginning. And if you look on this slide, you can see on the left side a typical plasmid. There is a dotted line, and everything on the left side of the dotted line is what is necessary to amplify the plasmid in E. coli cells, but what is, of course, not required for any pharmaceutical application, because the black part on the left side is the origin of replication. It only works in E. coli. And the yellow part is the ampicillin resistance gene. This is also only working in E. coli, but it shouldn't be in a human or in a producer cell. So it would be perfect if you could only use the right part of this plasmid, starting from blue, which is the promoter, the orange is the gene of interest, and the red part is the poly-A signal. So this gene of interest should be the one that we want to apply to the cell. And we placed it into our minicircle production system, which contains recombination sequences approximately at the site where the dotted line is crossing the plasmid. So we have 2 of these recombination sequences, and they make this 8-like structure, which you can see on the right side. And it is a recombination within the molecule. It's a cis recombination which is causing 2 circles. The upper one with the orange gene of interest is what we want to have, the minicircle, and the lower one is the mini plasmid. It contains simply those elements that we do not want to have in our product and that we want to get rid of. So this is the principal system of making them. It happens all during the cultivation. As you can see in the next slide, we start an induction at point 0. Before point 0, we have a lower and a higher band. This is a monomer and a dimer. Remember from the beginning that there is monomers and dimers existing. Then we start the induction, and after some minutes, already after 2 minutes, you can see a lot of minicircle and mini plasmid showing up. And after 10 minutes, this is almost completely done. No parental plasmid left over, only 2 bands. The upper band is the mini plasmid, the lower band is the mini circle. They are both there, and the parental plasmid is gone. And this is the recombination within the fermentation process. And from there, we harvest the cells. And what's next is that we have to separate now the non-desired mini plasmid from the desired mini circle. This is shown in the next slide. There is 2 agarose gels on the left side. You see M is the size marker of DNA fragments. You see that 1 and 2 is before the induction of the recombination, and lane 3 is the 2 bands, like you have seen them before, the mini plasmid and the mini circle. Then we do a purification process in between, which is a proprietary technology which is simply isolating the minicircle, and this is then leading to a minicircle, lane 4. And if we check for the quality of this again with the capillary gel electrophoresis on the right side, you can see the electropherogram of this. You can see a very clean CCC peak for the monomer of the minicircle, which is super coiled. This is what we want to have to use in our different applications. One was the one with the AAV viral particles. If we use such a minicircle, we don't have the antibiotic resistance element there, and we don't have the origin of replication. If we don't have it in our process, it cannot be false packaged into these AAV particles. The next slide shows you 4 different constructs. The upper left is the helper packaging plasmid, quite big, where we just changed the structure by making a minicircle out of it, not containing the origin of replication, not containing the antibiotic resistance. And the lower left is the transfer plasmid, the one with the ITR sequences, those which are used as packaging signals to make the AAV particles. The dotted line shows, like before, the part that we do not want to have, and also this is a minicircle on the right side, which does not contain the origin of replication and also does not contain the antibiotic resistance. So we have 2 minicircles then, instead of 2 plasmids. They are a little bit smaller now than the plasmids, and they are used for co-transfection. And in this overview, you can see that there is different columns, and they all show how many wrong packaged viral particles we could identify with qPCR. And if we combine the vector construct as a plasmid and the helper construct as a minicircle, we see this, as well as what I have presented before, plasmid and plasmid, but also minicircle and plasmid in the other combination shows still some wrong packaged viral particles, but less if the vector construct is a minicircle. And on the right side, where only minicircle and minicircle is used, the 2 that I have shown you in the slide before, only minicircle in the process, no plasmid at all, then there is also no ampicillin resistance marker at all, and this means the vector particles are completely free of such wrong sequences, which is a real advantage in manufacturing, because simply the batch cannot be contaminated by these E. coli sequences that shouldn't be in this product, especially not if it's a pharmaceutical product. And based on the idea of having a minicircle which does not contain elements which are deriving from E. coli and shouldn't be there, we got contacted from colleagues working with CAR-T cells, and they have all the problem that they work with lentiviral vectors, which is the technology that works today, and they all wanted to try using plasmids for making CAR-T cells, but it didn't work because plasmids seem to be more toxic than expected, and it turned out that it could be a reason that this toxicity is coming from the part of the plasmid that nobody needs. Again, the origin of replication and the antibiotic resistance. So the CAR-T cell therapy makes use of modifying a T cell being a CAR-T cell then, and this CAR-T cell is then fighting against cancer cells. There is an article of Michael Hudecek that I mentioned in the right corner that you should have a look at. And what we did was that we shifted the plasmids that have been used from the plasmid system to the minicircle system to find out if it works to go with plasmids. They are small, they go to the nucleus, and this was combined with the Sleeping Beauty technology of the MDC in Berlin. And this technology allows that a gene of making the CAR T cell from a T cell that this is jumping into the genome, and this jumping into the genome is done by the Sleeping Beauty technology. It's a transposon-like system, and on the next slide you can see that there are sequence elements right and left of the light blue element in the left plasmid, and this is what should jump into the genome of the T cell to make a CAR T cell out of this. And on the right side you see a plasmid that is carrying an SB100 gene. This is the transposase, and if this is expressed in a cell where the left plasmid is present as well, then the gene of the right plasmid is expressed, and the enzyme generated is causing the jumping of the light blue sequence into the genome, and it's randomly integrating, which makes it safer than integration into genes that can happen with alternative systems. And this is why people were interested in this, but it simply didn't work that good while it was based on a plasmid. And what we did, next slide, is we made minicircles out of it. They look like before. They simply don't have the origin of replication and the antibiotic resistance again. And these two were co-transfected to make CAR-T cells from T cells. And this was done in mouse experiments. And if you have a look to this time score here, you can see that the mice were treated with cancer cells. The tumor was growing until day 7, became bigger and bigger. And on 7, there was a treatment with the minicircles. And then the CAR-T cells were produced in the body of the mice. And what happened was that those mice not treated, this is the upper lines, they have to die after 18 days, while the others were rescued, as you can see in the lower line. And this is a comparison between technology that has been used with lentivirus, and in this case it is mRNA and minicircle. The Sleeping Beauty gene was encoded on an mRNA, and the jumping genetic element is on the minicircle. This shows, and it's also visible by showing pictures of the mice, that with mRNA minicircle, you see after 14 days, the lower panel, the mice are cured and free of the tumor that was visible on day 7. And with the lentiviral approach, it's the same result, while all the controls on the right side do not show this. So this was a second example of using minicircles. Minicircles are smaller than the plasmids, as you remember, by taking out the parts that are not required. And this is what I wanted to show you. Further reading is possible if you're interested in plasmids or minicircles. There is some literature available, and I thank you very much for having a look to this. And this was the presentation, and would like to open it for questions if you have some.
Thank you, Dr. Schleef, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have any questions you'd like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. Let's get started then. Our first question, Dr. Schleef, is there any size limitation for making a minicircle?
Yeah, thank you for that question. We think that there should not be any size limitation. But to be honest, we not really know, because the smallest one that we made was 503 base pair in size, which is quite small. There's not a lot of genes being smaller than that, but we never tried something smaller. And the biggest one is something that you have seen in the presentation. It is around 21 kb in size, which is quite big for a minicircle. So the term minicircle maybe not really fits anymore, but it's possible to do this, and we think that even larger DNA stretches can be put onto the minicircle system. So we are very optimistic that there is no size limitation, which is very important for those people that want to deal with systems treating genes in gene therapy, which are big, because there are certain genes a lot bigger than 20 kb.
Thank you, Dr. Schleef. And an audience member would like to know, what is the unique feature of your minicircle?
Yeah, the most important thing is that this type of minicircle is supercoiled. Supercoiled is what I explained in the beginning of the presentation, the only really functional structure of a DNA that is very active in gene expression because of this superhelicity. But we also can make sure that the minicircle is a monomer. Certain attempts to make minicircles in the past were working with systems that made multimeric forms of minicircles. They contained a lot of genetic elements coming from the system itself, and they are looking like a ladder on an agarose gel. So only the monomer is pharmaceutically perfect, because in pharmaceutical point of view, you need a homogeneous product, and homogeneous is if it's one band on the gel and supercoiled. And this is what I think is the most important feature of this type of minicircle.
And Dr. Schleef, are minicircle, are they clinically applicable?
Yeah, this is, I would say, the future of the minicircle. So far, as you have seen, this was applied in mouse models of a disease. And of course, the people doing this research and a lot of others that have read our publications on this were interested in testing their genes of interest in their disease models with the minicircle as well. So we made a lot of them already under research conditions, and they got extremely good results with that. But now they all ask for the next step, and the next step is how to go into curing a disease. And we have a special product here, which is called the HQ Lab. This is a high-quality production facility dedicated for products that can be used in applications where high quality is necessary, and this means there's only one product per time. There's a cleaning, there's a quality management system, there's SOPs, there's everything what people dealing with pharmaceutical starting materials like. And in this facility, we start from January on the first productions of minicircles in that high quality, so that it's possible to go with them further, to be able to manufacture starting from those molecules, either AAV or maybe other products that are then used in this high quality applications, clinical applications, maybe. This depends on the regulatory authorities. And this is the first step towards further quality improvements, where we, of course, also work on, but I cannot tell you exactly when this will happen.
Thank you, Dr. Schleef. And these are great questions from our audience members. It looks like we have time for one more question. Dr. Schleef, what are the requirements for DNA as a starting material for clinical grade AAV manufacturing?
The most important thing is that the starting material is produced in a documented way. And there I would like to come back to this HQ lab that I mentioned. There everything has to be traceable. This means all raw material, the facility itself, the machines and devices, all equipment, this all has to be controlled, and it should be monitored. And if this high quality of the environment is given, and if everything is documented, and if everything is traceable with respect to the raw materials, then it's possible to manufacture a product there which can be easily used in a GMP manufacturing process, and will be used to make a high quality viral vector, for example, or an mRNA, whatever people like to make, starting from the plasmid or in the future from the minicircle.
Thank you, Dr. Schleef, and I want to thank you for joining us today and for your important research. I'd also like to thank LabRoots and our sponsor, SciEx, for underwriting today's educational webcast. Before we go, I'd like to thank our audience for joining us today and for their interesting questions. Questions we didn't have time for today, and those submitted during the on-demand period will be addressed by Dr. Schleef via email that you provided at the time of registration. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share this email with your colleagues who may have missed today's live event. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share this email with your colleagues who may have missed today's live event. You will now be redirected to the registration page of our next SciEx webinar titled A Sensitive and Robust Plasmid Analysis Method by Capillary Electrophoresis with Laser-Induced Fluorescence Detection, presented by Colleen Reed, a scientist in analytical research and development at Pfizer. The webinar will broadcast live on December 11th at 7 a.m. Pacific time. We hope to see you there. Have a great day.